Hi, Dr. Lalitha. I'm really excited to have you on the call today. Before we start this fireside chat, can you please introduce yourself and let us know what is that you do at PI Health Sciences?
Yeah, thank you, Raghav. Yes, I'm Lalitha Vijayakrishnan. I head Discovery Biology and Translational Sciences at PI Health Sciences. I'm an immunologist by training with almost 25 years of experience in drug discovery, having been in the pharma sector for this long. I did my PhD in immunology, then moved to the US for a postdoctoral training at Harvard Medical School. I returned to India. I joined the pharma industry almost 20 years back and have been in the pharma industry ever since, working both on small molecules and large molecules in the integrated drug discovery space. So that's a brief about me. Thank you.
Doctor, to start this conversation, I want to ask you this question. PI Health Sciences being an integrated CRDMO player, what role does drug discovery biology and translational sciences play in drug discovery value chain? Can you expand a little bit on that?
Yeah, so let me speak briefly to that. Discovery biology and translational sciences, right, as the name of the department goes, plays integral part not only in the early part of drug discovery, but also towards the latter half, which is the translational aspect. So in the earlier phase, the biology team is in, the component of its contribution is mainly to screen drugs based on the mechanism of action and identify molecules that could potentially become lead candidates. That is, those are ones which can have all the properties that are required in terms of enzymatic activity and mechanism of action to take the drug forward, right? To take these molecules forward. As you know, chemists make hundreds of molecules and not all of them are really having the properties that are required to make it into a drug. So that is essentially what the biology team does, screens these compounds for chemical and more so for biological properties that are suitable for taking the drug ahead. And at the translational phase, what happens is that you now have molecules that are active both in vitro and in vivo, but do you really know how effective those compounds are in terms of treating patients, and how closely can you get to clinical relevance, right? Again, what biology does is using patient samples or tissues and cells derived out of patients which are related to this the target. It helps to identify whether the mechanism of action that has been identified in the in vitro and in the in vivo stages actually play out in samples which are derived from patients. So this is the extent of value chain that discovery biology plays in the entire process of the integrated drug discovery.
So, Doctor, my next question is, can you tell us what are the various challenges that you face in an early stage drug discovery programs? Can you elaborate a little bit on that?
Yes. So some of the early, it can be quite a long discussion, but to just put it briefly, some of the early challenges one would face is how good is your validation of the target, right? The molecular validation, if there is genetic validation or if there is pharmacological validation, that is very critical, because if you look at the attrition rate of these projects, right, a lot of them never see the light of day simply because targets have not been validated well enough at the very early stage. So I think that would be the most critical aspect that one has to consider before even venturing into a drug discovery program. And then post that are all the assays and the animal models that you have put in place to identify the right molecule. So if these assays and the animal models that you have are not exactly suited to addressing the mechanisms of action, then you probably are going to miss what you're looking for, because you then will identify those outcomes very late in the project, and that may be too late because a lot of time, money and manpower has already been invested, right? So these are some of the things that you need to have put in place well before you venture into initiating a drug discovery project.
So, Doctor, before I let you go, I want to know, what are the capabilities that make PI Health Sciences well positioned to take various discovery projects?
Right. So when you talk about discovery projects, I mean, there are multiple aspects to it. Anything which is preclinical, which comes preclinical, are discovery in stage, right? So in terms of early discovery, I think it is the infrastructure that we have, all our high throughput systems, in terms of machinery and equipments that we have. These are very well suited at the level of the throughput that we have to address and speed up projects, expediting their individual stages, that was from identifying the hit to lead optimization. All of those have to be expedited if you want to arrive at the the compound that you want to move ahead with. And I think our high throughput screening mechanism, a medium to high throughput, I would say, is well suited and is definitely a capability that we can be proud of. In fact, again, all our instruments, right, are 21 CFR compliant, IQ, OQ, PQ. So there is really a lot of credibility to the data that we generate. So these are the two aspects, I would say, some of the very important nuances that needs to be put in place before we start a project. And beyond that, I would think are the kind of technologies in terms of platforms that we have available, right? It is not only for small molecule screening, in vitro and ex vivo and in vivo. It's also about the larger molecules, the monoclonal antibodies, the bispecific and the trispecific, the engineered antibodies that we can screen, and more so in the kind of therapeutic areas that we can work. Oncology is one of the fields that we are very well versed with. And in oncology itself, our approach is not only through small molecules, but also through the more recent modalities like the PROTACs, the AutoTACs, the RIB-TACs. We are well versed with screening technologies that can support medicinal chemistry for all these modalities. In addition, is the fact that in oncology itself, we don't restrict to 2D screening. We also also are very familiar. In fact, we have a team which has a lot of expertise in development of 3D spheroids and organoids. Organoids specifically from patient-derived xenografts, patient-derived tissues. So we are able to develop 3D organoids as well. So all of these are capabilities and modalities that we have, which actually distinguishes us and differentiates us from CROs all over the world, I would say. So I would think that this is a very brief about our capabilities, but we are well head and shoulders well above the rest of CROs which all contribute to these similar services.
Dr. Can you elaborate a little bit more on immunogenicity studies and services that you are working on PI Health Sciences?
Yeah. So thank you, Raghav. That is a question which is more related to a recent portfolio that we have as a service offering at PI Health Sciences, where we address the immunogenicity of not only small molecule, but also peptide and monoclonal antibodies. Here, a recent mandate by the FDA requires that generic peptides need to be shown to have bioactivity similar to reference listed drugs in terms of them being their immunogenic potential, right? So we have developed what is the innate immunogenicity study and the adaptive immunogenicity study. Both these studies use primary cells derived from human whole blood. We isolate PBMCs and evaluate both these drugs to determine comparison and safety. So this is a recent portfolio and well aligned with what are the FDA requirements for these studies. So this is to sum up all our services. But what I would like to say in closing is the fact that this conversation is not exhaustive enough to cover all our capabilities. We do have more in our service portfolio, and we request that you reach out to us to understand our services better, and also in case you would like to follow up on certain of what has been covered in this conversation, please feel free to reach out to us. Thank you.
This is really very interesting, and I know we're just touching the surface with this conversation, but I really want to thank you for giving your time for us, and I assure you this is not going to be the last of it. We would definitely have more conversation with you to learn about what you're doing and what you're driving and what are the various things that's happening at PI Health Sciences. Once again, I want to thank you for your time, and thanks a lot for taking your time out for us.
Thank you, Raghav.